Okay, so what we're going to look at now is genetic technologies and genetic engineering in particular. We're going to cover recombinant DNA technology, have a little bit look at that in a bit more detail, and then do some exam questions based on recombinant DNA tech. So that's part of the genetic technologies topic that we're looking at right now. So in this session, what we're going to look at is genetic engineering, how we use it, what recombinant technology is, learn about producing DNA copies, understanding bacterial conjunction and conjugation and recombinant bacteria, and then about other genetic markers and viruses as vectors. So how is genetic engineering actually useful? So we had a little bit of talk on the last session, where we were looking at cloning, about how clones would be really useful. But genetic engineering is different to cloning. We're not just making an exact copy, but genetic engineering is actually where we insert a foreign gene into another organism's genome, and it results in the expression of a new gene. So you'll see sometimes where you can get glow-in-the-dark mice, and that's from when we've used the phosphorescence that you can find in animals such as squid and jellyfish, and we can take them out of them and take that phosphorescence and pop it into the mice, for example. So this method can be used to engineer recombinant organisms, and they synthesize useful products such as hormones. So actually, if we want certain hormones being made, we can actually pop that gene into another organism and it'll make those hormones, and we could harvest those hormones and use them somewhere else. But we can also use it to improve a feature of the recipient organism, so producing herbicide-resistant crop plants. So we have those two different ways we can look at it. We can look at it as making a recombinant organism, and we can use something that's being made in an organism that we wouldn't normally have and use that elsewhere, or as a recipient organism. So genetic engineering technologies aid the understanding of how organisms function by allowing scientists to study and alter gene function. So recombinant technology. So once the gene for the desired protein has been identified, we have to go through a few steps. So the first thing that people would do is decide, well, what is the gene that we want? What is the desired protein we need? Where is that gene on the genome? And we find it. Once we've identified it, we have to go through these steps. We need to make multiple copies of the desired gene. We can't just make one copy, we need multiple. We then take this gene, we insert it into a vector, and then we transfer it into host cells. The host cell that's successfully taken up the gene are identified, and we use a marker to help identify them. And then the host cells are allowed to multiply, or they are then cloned. So that's a really, really basic overview. We're going to look at that in more detail in a bit, but that's just a really, really nice, simple overview. But we'll look at how we can actually make these DNA fragments in a bit more detail in a moment. So producing DNA fragments using reverse transcriptase, and that's what we're going to look at now, is how we make these little copies. We'll have a little talk about genome projects as well. So when we make these DNA fragments that we're going to go and use for recombinant DNA further on, we make them using something called reverse transcriptase. Most cells only contain 2 copies of each gene, so it makes it really, really hard to obtain a DNA fragment containing the target gene. But our cells contain many, many mRNA inside them, and these mRNA molecules are complementary to our genes. So the mRNA is actually easier to obtain. So when we make these DNA fragments, we're going to be using mRNA because there's lots of mRNA inside the cell and they're easier to obtain. Quick reminder that because we're dealing with mRNA at this moment, we have uracil instead of thymine present. So that's something just to keep in mind. So we get the mRNA. So for an example here, we might want beta cells in the pancreas, and they contain a large amount of insulin mRNA. If we actually went straight to the nucleus, it would just have the 2 copies of it and it wouldn't be enough. So we go and get the mRNA. And what we can basically do then is we use these mRNA molecules as a template. And we're going to use these mRNA molecules to make lots and lots of DNA. And we're going to use the enzyme reverse transcriptase to make this DNA from the RNA template. And the DNA that we're going to make is going to be called complementary DNA. So this might be something that you've heard of before, maybe not. Just lost my pen though. It's called complementary DNA, sometimes written as cDNA. So cDNA, and that is complementary DNA. So that's the DNA that we're going to make from this RNA. So those pancreatic cells, we'll use this one as the example. It's one that you'll see used a lot in lots of places, produce the protein insulin. They've got loads of mRNA inside them that's complementary to the insulin gene, but only 2 copies of the gene itself. So what we're going to go along and use is reverse transcriptase to make that cDNA from the insulin mRNA. So what we have to do is we first need to isolate the mRNA from the cells. Then we're going to mix it with DNA nucleotides and reverse transcriptase to get this cDNA. So we have to isolate it first. So the mRNA has to be isolated. And so, for example, we could isolate it by breaking the cells open and centrifuging the contents. So break it open, centrifuge it, and basically we are just giving it lots and lots of force and that force is just going to break open those cells. It's nothing really fancy. We just break the cells open using base and then centrifuge them to really, really separate all those contents that's inside. So that's how we can isolate that mRNA. Then this is actually the making of those DNA fragments, those cDNA fragments using reverse transcriptase. So then what we go along and do is the enzyme reverse transcriptase and a supply of free DNA nucleotides are added to this test tube. So this test tube where we basically got all the mRNA in it and we've broken apart this cell, we're going to add reverse transcriptase and some free DNA nucleotides to it. This will then result, so we add all them in, and this is going to result in the synthesis of a single strand of complementary DNA using the extracted mRNA as a template. Now, what's really important is that this is all specific to that insulin, if we use an insulin example, that insulin mRNA. And that's what happens there. Then the final part is another enzyme, which we call ribonuclease H, is then added, and that breaks down the mRNA strand and just leaves us with that single stranded piece of DNA, that cDNA. So that is one way, and there's actually multiple ways we can do this. So that is one way of producing DNA copies. So DNA fragments, sorry. So that's one way we can make a DNA fragment, and that's using reverse transcriptase. There is another way we can make DNA fragments. We can actually cut our DNA. And we can also use a gene machine. I'm not sure if gene machine's covered on here, so I will do a bit about that in a moment, if not. And then finally, oh, sorry, I forgot this little bit. DNA polymerase and DNA nucleotides are added. A complementary strand of DNA is produced. We've already done this. The result is a fragment of double-stranded DNA. That is a copy of the original, but without the introns. But we don't need to worry about that bit, the AQA. So how do restriction endonucleases work? This is a different way of making DNA fragments. So restriction endonucleases are enzymes, and we use them to cut DNA into smaller fragments. And what these enzymes do is they catalyze a hydrolysis reaction. And what that hydrolysis reaction does is break that phosphodiester backbone of the double-stranded DNA in different places. And this can result in some exposed bases known as sticky ends. And what we're going to do now is look into the differences of what happens here. Okay, so restriction endonucleases, EcoRI, that's the shorthand for it, and we'll look into that in a little bit more detail. EcoRI is restriction endonuclease, and it recognizes the base sequence GAATC from the 5' to the 3' end on DNA. This enzyme basically it comes along and it cuts between the guanine and the adenine. I'm going to explain this a bit more. So some sections of your DNA have a palindromic sequence of nucleotides, and a palindromic sequence is when these sequences consist of anti-parallel bases. So I'm going to draw them out here so you can see them a bit better actually by what I actually mean. So this GAATC, if I draw this here, I draw an example of our DNA. So we could have C, and I'm going to do that G, A, T, T, C here, and then I'll just stick a G and an A on the end. Obviously, that's going to base pair with G, C, T, T, A, G, C, T. I'm going to highlight which area is a palindrome. So the palindrome is that GATTC. And it's a palindrome because when we read it in this direction, which is our 5' to 3' direction, this way, it reads GATTC. And when we read it in this direction, which is our 5' to, when we read it in that direction, we've got GATTC. So the sequence consists of anti-parallel base pairs. So these are base pairs that read the same in opposite directions. So when we read them in the opposite directions, they read exactly the same. So this is our palindrome sequence. So restriction endonucleases are enzymes that recognize specific palindromic sequences. These are known as recognition sequences. And what they come along and do is they digest the DNA at these places. So you can have a whole load of different restriction endonucleases, and they will cut at different specific points to specific recognition sequences in your DNA. And that's because the shape of a recognition sequence is complementary to the enzyme's active site. And I'll repeat that again because that's really important. So different restriction endonucleases cut at different specific recognition sequences. And that is because the shape of the recognition sequence is going to be complementary to the enzyme's active site. So the example that we've got on here, if I clear all my drawings off now, is EcoRI, and that cuts at GAATTC. A different example would be Hind3, which is spelled like this, Hind3, and that cuts at AAGCTT. So you don't need to know them, it's just some examples there for you. So EcoRI recognizes the base sequence GAATC from the 5' to the 3' end on DNA. This enzyme cuts between the guanine and the adenine. Oh, we haven't been able to see that very well. That would have been better if we'd seen that there. And I'm going to replay that if it'll let me replay it. We couldn't see all of it. Let's try that now. Nope. Okay. So you then get two ends being left, which are called sticky ends. And I'm going to explain them in a minute, but I'm going to just show you it with Hind3 as well, because I'll scroll it down so we can see that a bit better. So if I go back here, is there any way I can put it on? No. Oh, yes. Okay, here we go. So this is our EcoRI cutting between the guanine and the adenine. And then it does exactly the same on the other strand of the DNA. When it does that, it leaves 2 points sticking out the end, and we call them sticky ends. So the TTC, sorry, is the recognition sequences. We don't want them. So basically they get cut out, and all this stuff in the middle, and when I say stuff in the middle, this stuff here is the stuff that we want. So this is the fragment that we want. This is what we want. These would have been our recognition sequences that we've got rid of. When the EcoRI cuts in, so basically if recognition sequences are present, which they were, at either side of a DNA fragment, you can use a restriction endonuclease to separate it from the rest of the DNA, which is what it's done. The DNA sample is incubated with a specific restriction endonucleases. And what they do is they cut the DNA fragment out. And as I said, that's using that hydrolysis reaction to cut that fragment out. Sometimes the cut leaves what we've got here, something called sticky ends. And these sticky ends are small tails of unpaired bases at each end of the fragment. Sticky ends can be used to bind, or we can use this other word, anneal, the DNA fragment to another piece of DNA that has sticky ends with complementary sequences. So we look at that a bit later on, so we won't cover that right at the moment, but that's what we can do with those. Let's try now to go on to the next part. So which restriction endonuclease would be used to cut vector DNA so that the DNA fragment can be incorporated into the genes? Well, we'd want to use one that would give sticky ends. So hind 3, as you can see here, how that's going to cut, is going to have sticky ends. And we want those sticky ends because they're going to be complementary. What we want is sticky ends that are going to be complementary to those base pairs. That hind 3 cut, but didn't give us complementary base pairings to that vector DNA. So hind 3 wouldn't work. What I'm really hoping is it's going to carry on playing and give us one where it would be complementary. So here we go, we look at another one. So we're going to use tack 1. It's going to cut where it needs to cut. And we're going to have a look now if they're going to be complementary to the vector DNA. They're non-complementary base pairs, so we wouldn't use those ones. And then finally, EcoRI. They're complementary, so they're the ones we choose for that vector DNA. And that's because it's a sticky end, and then they anneal to that vector DNA to attach that into the vector. So which process does the statements refer to? Producing DNA fragments using restriction enzymes, or producing DNA fragments using reverse transcriptase? Uses DNA polymerase, and that is reverse transcriptase. Results in cDNA, that's reverse transcriptase. Cuts DNA from a genome, restriction enzymes. Produces fragments with sticky ends, restriction enzymes. Contains introns. Now, this one's a bit of a debatable one. So I'm just going to pop contains introns over here for a minute, and we'll come back to it in a sec, so I'm going to move it that. Uses mRNA as a template, reverse transcriptase. Contains introns. We're going to pop that under using restriction enzymes, because in the reverse transcriptase, we would then remove the introns, whereas using restriction enzymes, we're not getting rid of introns. So we're going to still have those introns there. So we have something called bacterial conjugation. And we've used this knowledge actually to help us a lot with genetic technologies. So microorganisms can naturally exchange genetic material, and that's in a process called conjugation. Genetic material in the form of plasmids can be copied and passed between bacteria. So some plasmids can contain genes associated with antibiotic resistance. And as those plasmids move between individuals of the same and different species, it speeds up the spread of that antibiotic resistance. So what we're going to have a look at now is recombinant bacteria. Now, I'm saying this, but I'm just going to pause this for 2 seconds because I've just given you 2 ways of making DNA fragments, but there's one other way of making DNA fragments that I just want to go through. Okay, so the 3 ways of making DNA fragments are, as I've said, 1, using reverse transcriptase. Let me use this pen, this one would work better. Transcriptase. The second is using restriction endonucleases. And then the third one, which is what we're going to go through now, is using something called a gene machine. So that's the last bit. So more recently, the gene machine is the new one. Oh, we've just got a random word reverse flying around. Gene machine. More recently, technology has been developed so that fragments of DNA can actually be synthesized from scratch without the need for pre-existing DNA template. So this is made from scratch. We don't need that pre-existing template. So instead, what we have is we have a database, and that contains all the necessary information to form that DNA fragment. So we use a database. Oops. We use a database to get all that information. So this means that our DNA sequence does not have to exist naturally. So we can make any sequence. So it could be one that doesn't actually exist in nature at all. So we can make any sequence. So this is how it works. Gene machine, we make it from scratch using database, and we can make any sequence. So the sequence that is required is designed if it doesn't already exist. If it exists, we just use the one that already exists. If not, we design it. So number 1, what we would do is we design the sequence. Then what we do is the first nucleotide in the sequence needs to be fixed to something. So some sort of a support. So our first nucleotide in the sequence, is fixed to something. We often use a bead. I'm going to use that as this example. The first nucleotide in sequence is fixed to a bead. Then we just go through and add each nucleotide step by step. So we just go along and we add them all step by step in the correct order. And it's in a cycle process that includes adding something called protecting groups. So we add these protecting groups. And protecting groups, what they do is they actually make sure that the nucleotides are joined at the right point. So these make sure nucleotides are joined at the right point. Step 4 is our last point. So then our short sections of DNA, and these are called oligonucleotides. And these are 20 nucleotides long, are produced. Once these are complete, they're broken off from the support, all protecting groups are removed, and then they can be joined together to make a longer DNA fragment. And then we join to make a longer fragment. So that is the gene machine and how the gene machine works. I'll quickly go through that again. We design the sequence if it doesn't already exist. First nucleotide in the sequence is fixed to a bead. Each nucleotide is then added step by step. We add protecting groups, which make sure nucleotides are joined at the right points. And then at this point, we've then formed oligonucleotides, which are roughly 20 nucleotides long. At this point, we remove the supports like the beads and the protecting groups, and then we join all the oligonucleotides together to make a longer fragment. Okay, so coming back to recombinant bacteria now, we can use plasmids as vectors. So plasmids are circular lengths of DNA that we find in bacteria, and these can be used to transfer recombinant DNA into bacterial cells. So what we would do is a gene for the desired protein, we're going to stick with insulin, and this can actually be incorporated into a plasmid. So what we're going to do is that specific restriction enzyme used to cut the gene, we actually also use it to cut the plasmid. So we've made our DNA fragment, and actually we're going to use that restriction enzyme, same one, to come and cut the vector in the same place. So then we've got matching sticky ends. So our desired gene can fit straight in. That desired gene is then inserted into a plasmid, and we use the enzyme ligase to join the sugar phosphate bone together. It's likely that not all the plasmids will take up the desired gene, but that's fine because inside of the bacteria, we have lots and lots of plasmids. So as long as the majority do, that's great. When we use these plasmids containing antibiotic-resistant genes, it can allow the bacteria containing the desired gene to be easily identified. So the new gene is inserted into the middle of one of these antibiotic resistance genes. So that's really good. It makes it really easy for us to actually ID which individuals of the bacteria have got this recombinant DNA inside them and which ones haven't, because not all of them are going to take it up. So it's not really that useful if we can't actually find it again. So we have to have a way of identifying it again. So bacteria and our plasmids are mixed together. And what this does is it allows the bacteria to take up the plasmids. Plasmids can pass through the cell membrane. Only a very small number of the bacteria will actually take up the plasmid. So again, that's why we put the gene into where we have that antibiotic resistance, because then when we grow the bacteria, the ones that are antibiotic resistance, we can use that to help us ID if it's got the actual vector taken up, the recombinant DNA. So bacteria are cultured on agar plates that contain ampicillin, because ampicillin was one of them that they had resistance to. And then small samples of these bacteria are then grown on tetracycline. And can you guess which colony on the original plate contains the desired gene? So B is the bacteria that contains the desired gene. This population contains the ampicillin resistance gene from the plasmid, but they're not resistant to tetracycline as the gene was interrupted by the inserted gene. So that one shouldn't work. So that is the one that we know is definitely the one. So the transgenic bacteria can then be grown on a large scale in order to produce a large amount of the desired protein, so the insulin. So we then take that one bacteria and it will then go through growth so that we can then get lots of them. Other ways that we can work out if a host is holding our recombinant DNA is using fluorescent markers and enzyme markers, and we can do things like DNA probes as well. So these are techniques to identify cells that have taken up the desired gene. So a gene from a jellyfish that produces a protein called green fluorescent protein, GFP, can be used. And so the desired gene is then transplanted into the center of the GFP gene, and the cells that haven't taken up the desired gene have not taken up the desired gene will fluoresce, whereas the others won't. So the ones that don't fluoresce are the ones that we want, and we can get rid of the ones that fluoresce. We can also use enzyme markers, and these involve transplanting the desired gene into the center of a gene coding for lactase. Lactase will turn a particular colorless substrate blue. Therefore, the cells that have taken up the desired gene will not turn this substrate blue. We can also use viruses as vectors, and that's because viruses naturally transfer their genetic material into their host cells. And because of this, they need the host to produce viral proteins to allow them to replicate. Scientists use viruses such as the adenovirus as vectors in DNA tech. So the genetic material that causes virulence must first be removed from the virus. So we have to take that out, and then we can add the desired gene to the viral genome. The virus then infects target cells, inserting its genome so that the target cells then express the new sequence. So enzymes for processes, reverse transcriptase is the production of cDNA fragment from an mRNA strand. Restriction endonuclease is recognition and cutting of DNA at specific base sequences. DNA ligase is binding of new DNA fragments, and DNA polymerase is formation of new DNA strands during DNA replication. Okay, so what we're going to go through now is we are just going to go through some exam questions, looking at recombinant DNA. So we're going to go through that now. If for some reason there is a little bit this section missing about recombinant DNA, I will be putting that onto the video after this, and because I might have deleted it by accident. So we will find out in a moment, but don't worry. If this video suddenly jumps from making DNA fragments to then talking about recombinant DNA, and I haven't talked about recombinant bacteria and recombinant viruses as hosts, there will be a video to follow, and you'll just need to sort of watch up until I start talking about this, watch the other one, and then come back to this one. So I'm sorry about that. I don't know what's happened. But I might also be talking about nothing, and it might all be fine. So there you go. So basically, this first question, and so some populations of flies are becoming resistant to insecticides which intend to kill them. Scientists developed a method for finding out whether a fly was carrying a recessive allele or that gives resistance to an insecticide, or the dominant allele that does not give resistance. So what scientists did is they crossed flies with the homozygous dominant allele with flies with the genotype of the recessive allele. They obtained DNA samples from the parents and offspring, and they used the same restriction endonuclease enzymes on each sample to obtain DNA fragments. So the first question is, explain why the scientists used the same restriction endonuclease enzymes on each DNA sample. Well, if we're going to obtain the DNA fragments, we want them all to have all the DNA fragments to be exactly the same. If we use different restriction endonucleases, we'd have different fragments. So we use the same restriction endonuclease enzymes to make sure that we cut the DNA at the same base sequence. Therefore, we will get fragments with required gene. That would get you 2 marks. The next part of this question goes on to say, the scientists added 2 different primers to each sample of DNA for the polymerase chain reaction. So we're going to look at PCR. So primer A3 only binds to 195 base pair fragment. Primer A4 only binds to 135 base pair fragment. The scientists separated the DNA fragments produced by the PCR in a gel, where the shorter fragments move further in a given time, and the results are shown. So we've got these. And what this asks you to do is explain why primer A3 and primer A4 only bind to specific DNA fragments. So primer A3 the reason that this happens is because each primer has their own specific base sequence. And that is complementary. So each primer has a specific base sequence that is complementary to the allele. You're then asked to explain the results in figure 1. I don't want to spend a lot of time on that actually at the moment, because this isn't something that we're looking at, and we're focusing on recombinant DNA tech at the minute. So we'll skip past this part for the moment. And we're actually going to leave the rest of this question for now. We're going to look at question 2 though, where we look at silkworms. So silkworms secrete silk fibers, and these are harvested and used to manufacture silk fabric. Scientists have produced genetically modified silkworms that contain a gene from a spider. The genetically modified silkworms secrete fibers made of spider web protein, spider silk, which is stronger than the normal silk fiber protein. The method the scientists used is shown in the figure below. So they isolated the gene from the spider. They inserted the spider gene into a plasmid with a marker gene. This was called EGFP. This gene codes for a protein that grows brightly in ultraviolet light. They inject copies of this plasmid into many eggs of silkworms and allow silkworm eggs that have taken up the plasmid to grow and produce spider silk. First question for this is, suggest why the plasmids were injected into the eggs of silkworms rather than into silkworms themselves. I feel like I've missed a question up here. So we're going to inject it into the egg. The genes will go into most of the cells of the silkworm. If we inject it into the silkworm, the genes won't be spread as far. So suggest why plasmids injected into the eggs of the silkworms rather than into the silkworms, because in this situation, our genes will enter all of the cells. All of the cells, most of the cells, most. So we can guarantee it, so it gets into cells that will make the silk. Otherwise, what could end up happening is if we just inject it into the silkworm, it doesn't go to the right place and it makes no difference. So then the next part of the question asks, suggest why scientists use a marker gene when they use the EGFP. Well, we know from what we've just learned that not all of the eggs, will take up the recombinant DNA, take up the plasmid. So not all eggs will successfully take up the plasmid. So we use the marker gene and the EGFP gene because that will mean that silkworms that have taken up, so silkworms that have taken up the plasmid will glow. So the scientists ensure the spider gene was expressed only in cells within the silk glands. What was scientists have inserted into the plasmid along with the spider gene to ensure that the spider gene was only expressed in the silk glands of the silkworms? That, as you know, is a promoter region. So they'll insert a promoter region to ensure it's only expressed in those cells. So suggest 2 reasons why it's important that the spider gene is expressed only in the silk glands of the silkworms. Now you're suggesting, you're not expected to know the answer, but anything reasonable. First of all, so we can harvest that protein. We're not going to be able to get to it unless it's being expressed in the right place. So we can harvest the protein, would be one. And another thing is those fibers, if they were present in other cells, might actually cause harm to the silkworm. And the last question we're going to go through question 3 now, looking at this one. So scientists wanted to measure how much mRNA was transcribed from an allele A of a gene in a sample of cells. This gene exists in 2 forms, and the scientists isolated mRNA from the cells. They added an enzyme to mRNA to produce cDNA. Name the type of enzyme used to produce the cDNA. As we know, if it's using this process, it would be reverse transcriptase. It then goes on to cover PCR, and I don't want to look at PCR at the moment, and then DNA probes. So again, I just want to leave the rest of that question for the moment, and we'll look at that probably next week. Okay, and we'll look at this one. So haemophilia is a genetic condition in which blood fails to clot. Factor IX is a protein used to treat haemophilia. Sheep can be genetically engineered to produce this factor in their milk, produced by their mammary glands. Diagrams show the stages involved. So the first stage, the gene for that factor is cut from the human DNA. Jellyfish gene is attached to the human gene. A promoted DNA from the sheep is attached. Copies of this DNA are then inserted into the nuclei of body cells from the sheep. Each nucleus is transplanted into a sheep egg cell from which the original nucleus has been removed. And then the egg cells divide to form an embryo. Each embryo is implanted into the uterus of a different sheep. So the first question is, name the type of enzyme that is used to cut the gene for factor IX from human DNA. And that, as you know, for cutting is restriction endonucleases. So then we get told the jellyfish gene attached to the human factor IX gene, stage 2, codes a protein that glows green under fluorescent light. Explain the purpose of attaching this gene. Well, the point of this gene is to act as a marker. And it's going to act as a marker gene, and it's going to act as a marker gene to show that the human gene has been taken up, or our recombinant gene, our expressed gene. So as a marker gene to show that the gene has been taken up. I'm going to put the desired gene here. So that'd be one mark. Second part of the mark. So the colour will only produced. So only the cells that have got this implantation, the embryos will fluoresce. So only embryos with the gene will fluoresce. That'll get you 2 marks. And we're going to look at this promoter region section in a bit. So what we're going to do is we're going to stop this session here, and we're going to look a bit more into DNA probes and promoter regions in the next session.